while I do think that automation, for example, is a good tool to overcome some of the hurdles that we see in ATMPs, I don't think that it is the ultimate solution for advancing CGT development at this point in time. It is probably good for future iterations, but I think when we look at ATMPs and the complexity of these processes, automation usually thrives in standardized processes that have minimum variability.
Welcome to the Smart Biotech Scientist podcast. I'm your host, David Brühlmann, and today we're diving deep into the fascinating world of advanced therapy medicinal products. I'm thrilled to welcome Oliver Kraemer, who is leading the technical development at a new biotechnology venture within Flagship Pioneering, who brings a wealth of experience from working with both biologics and advanced therapy medicinal products at industry giants like BMS, Sanofi, and Boehringer Ingelheim. If you've ever wondered about the crucial differences between developing biologics and cell therapies, or what it takes to navigate the complex landscape of ATMP development, you are in for a treat. Oliver will share insights from his journey that took him from traditional biologics to cutting-edge cell therapies, including both iPSC-derived and donor-derived approaches. Stay tuned for part 1 of this insightful conversation. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Oliver. Thank you for joining us today.
I'm very excited to be here. Thanks for having me.
Oliver, share something that you believe about bioprocess development that most people disagree with.
That's a good question to start with. I think there are a couple of things that might be a little bit controversial. So my passion is, of course, about ATMPs. So this will focus probably a lot on the ATMPs. One of the things that I think that is controversial is that while I do think that automation, for example, is a good tool to overcome some of the hurdles that we see in ATMPs, I don't think that it is the ultimate solution for advancing CGT development at this point in time. It is probably good for future iterations, but I think when we look at ATMPs and the complexity of these processes, automation usually thrives in standardized processes that have minimum variability. And CGTs, what we have seen is that they often require a lot of customization and they are very finicky. And if you look at CAR T processes, for example, or viral manufacturing, they don't always perform as predicted or as desired. And that's why I think that even a lot of people think that automation will drive the costs down, will make manufacturing more accessible. I think that is something that is not in the near future. So that might be controversial, but that's one thing that I have seen in my career so far. And the second thing that might be controversial is that I do think that the strict separation of research and development does not work that well for CGT. And we can elaborate on that later, why I think this is true and what the hurdles are. But I do think that it is very challenging to do CGT development or ATMP development with the current established models.
Great. So before we talk about the nitty-gritty of cell and gene therapy, let's talk about yourself. Oliver, can you draw us into your journey into the biotech world and highlight the key moments and decisions that shaped your career path?
Yeah, absolutely. So I will start at the very beginning. I was always very interested in biology, genetics. So when I was still very young, I always had this desire. I want to study something with cells and genetics. So it was not very defined at that stage in time. It was just something with that. And I was then evaluating different universities and thinking of what do I actually want to do with that. And that was actually the first time when my attention was drawn more to bioprocess engineering and the engineering aspect of it. And I admit I didn't know it I didn't knew it at that time, but that was quite an important decision for me or for the career path that I have taken to incorporate more of these engineering aspects that are quite useful, especially if you think about processes, manufacturing, scaling. All of that is very impactful. So that was one of the key moments or the key decision points that I had, not knowing that it was one. So that was interesting. The university that I chose to study back in Germany was quite industry focused. So a lot of the processes and the research topics we were working on were industry focused, which was a coincidence, but also very helpful for getting more closer towards industry and what you can actually do with biotechnology when you apply it in that setting. There was this time, which maybe every PhD student has, I don't know, but I was pondering, should I start up just my own company or not? And in the end, I decided not to, which was in hindsight a good decision because I went directly to industry. I started at Boehringer Ingelheim, my first job, and I learned so much there about how large biopharma works, how certain regulatory aspects work. And I think that was great because it broadens your perspective so much. You often don't know what you don't know. And so that was a great journey for me to just see there is so much more than just the more narrow field of cell engineering that I have been working on to bring a drug to the market or even to clinical phase. And so that was great. I worked then more on upstream process development for Sanofi. And Sanofi also had a cell therapy product, and I was working on that. And that rekindled my love again with, I want to work closer with the cells. So I made the decision to go back into cell and gene therapy or to work closer in that environment. And yeah, that brought me over the big pond to the US. And I started working technically for Celgene, but then it was merged into BMS. So I worked then for BMS in autologous and allogeneic cell therapy, then a smaller startup company, Tune Therapeutics. And now I'm at Flagship Pioneering and working fully in cell and gene therapy.
Yeah, that's exciting. And like you, I also enjoyed a lot starting in a big biopharma. I worked at Merck in Switzerland and I learned so much. And I think there's a lot to see and discover being in a big company, especially at the beginning of your career. Now, you have worked on both biologics and cell therapies. And for our listeners who are not so familiar with either one, let's dissect this first. So let's look at what are the key differences, and especially looking at the development processes for biologics and ATMPs, and how do these differences impact the timelines?
Good question. I think when you look at ATMPs are significantly more complex than biologics, for example. In the past, people have compared biologics to small molecules, and so there are a lot of good comparisons where you have, ascorbic acid or so as a molecule, and you show the size of that molecule, and then you have an antibody, which is one of the main products in biologics. And you already see that the complexity of this molecule is significantly greater. Now imagine that as a cell, for example. A cell as a living entity has hundreds of thousands of pathways that are active, that are regulated in certain ways and react towards the environment it is in. So it is a living drug, and therefore working with it, generating it, manipulating it, but also making it safe for a patient is quite complex. So while ATMPs are a subsection of biologics, they are significantly more complex. And I think that is one of the key differences that you have if you compare these two. The other thing is, that's something that I also like to highlight. When we talk about biologics right now, we often forget that biologics started quite a while ago in the 80s. And so we're looking at 40 to 50 years, probably even a little bit longer, of development time and research time that went into these. So there's a lot of history and a lot of knowledge that was built up over this time. ATMPs are still very new. So when people compare them, they often use a perspective from today where we say we have so much control over biologics and they're relatively easy to produce. And that's not the case with ATMPs. Well, of course it is not, because ATMPs are still quite new. And so you don't have this benefit of an existing platform where you can just crank out molecule after molecule, or gene therapy or cell therapy after another, just changing the target or so. Every product that you develop in ATMPs is pretty unique. So you face different manufacturing challenges. You face different regulatory pathways or challenges. For some of that is not established. So you have to restart these conversations and think about a lot of hurdles that you might encounter there. And I think the last point that is also underappreciated is the question of supply chain. So if you look at biologics, again, the majority is a MAP or so, a monoclonal antibody. They can be in different formats. They can be lyophilized. They can be in solution, frozen, chilled, whatever. But that is something that we have figured out over time. For ATMPs, there is the question, how can they be supplied? Do they need to be stored? Do they need to be stored, let's say, unusual conditions? So something that is a lot colder than -20, for example. Is that something that is available everywhere? Can you just ship them out easily? So the access to the patient is very different. But also on the other hand, the supply chain for your raw materials might be different. A lot of the components that you use in ATMPs are based on research grades. So it might actually not be something that is readily available for you to manufacture in GMP. So I think those are the key differences that I see between biologics and ATMPs, which also make ATMPs a lot more complicated to work in.
So because of the lack of a platform approach and obviously the much higher complexity, do we need to start out completely differently every single time we have a project? Or how does this impact the predictability or the transferability of knowledge we've gained from one product to another?
Well, first of all, when we say ATMPs, even though it's a subsection, it is a relatively broad field. It includes cell therapies, it includes gene therapies, and cell and gene therapy, even though they are always named in the same context, they work quite differently. So if you look at the gene therapy, there are things that you can learn from your previous product or your previous experience. For example, in the delivery, is your delivery viral based? Then you know already a little bit about that virus, about how it behaves when you manufacture it, what the downsides are when you go into GMP manufacturing or so. Those are things that help you in understanding what do I have to watch out for. Now, for cell therapies, that might be more complicated because we say cell therapy, but also cell therapy is split into very different things. So you have, of course, the thing that is very famous and probably people know most about are CAR-Ts. And the CAR-Ts are currently usually autologous ones. So if you have autologous ones, you have an inherent variation of your starting material because they are coming from the patient. So you have actually a little less control over that because you get what you get. And that makes the development there a little bit more complicated. Now, when you talk about allogeneic products, then again, the question is it a healthy donor derived, which is still patient material? You don't have unlimited resources of that. So you have to take that into consideration. But these products will behave different compared to the autologous ones. And then you have the whole stem cell field, which starts with ESCs or iPSCs or other forms of stem cells. And they, again, have very different challenges associated with that. So I think it is difficult to say, can I transfer knowledge from product one to product two? You can to a certain degree if there are enough similarities between your starting points. But in many cases, that is just not true. So yes, unfortunately, you have to look at all different aspects of the product development for each product individually. That will change over time. I mean, it changed also for biologics, and there will be a time where it will be easier to do this, but we're still a little bit away from that.
This means for now, we have to make a lot more decisions early on because we cannot rely on a platform approach.
Correct.
So how does this impact then the development strategy?
That's a great question, and that ties back to the point that I initially made. If you look at how the decision workflow is in classical biologics, you have the research phase, which looks a lot at, for example, target identification, lead identification, optimization of that. But then you have something that is bundled and can be transferred to your development unit. And then you can think about everything that is manufacturing associated. You can scale up the product, develop your process, everything that is necessary and associated with that. But for ATMPs, and I specifically enjoy, of course, talking about everything that is cell therapy related, that does not work that well. So if you look at, for example, an iPSC-based therapy, the question that needs to be addressed a lot earlier is very development and very manufacturing related. It starts with your cell line. Which cell line are you using for developing this product? Because if you compare the different iPSC lines, there are quite a bunch out there, but actually not as many as people would think. Some of them are just available for research use only. So you will never have a chance to do a clinical trial with that. Do you use that for your research work? Yes or no? That is an important question because everything that you develop is somewhat specific for that cell line. So when you later on have to change this because you use a line that is available for clinic and commercial, you will see differences and that will impact your development timeline. So you can make the case that you should actually prepone this decision and start with the evaluation of different cell lines earlier and just make this decision a lot earlier in the process. Same is true for the manufacturing process itself. How do you later want to supply the market or even the clinic? Is that a 2D system that you're using? Is that a 3D system? What about the media and cofactors that you're using there? All of these can impact the quality of the product that you're generating and thereby skew your research results in one direction or the other. Now, don't misunderstand me. I don't want to say that you have to do all of the classical CMC development work at first and then think about the product. That's not what I'm saying. I'm just saying that these aspects are just much more interconnected for ATMPs than they might be for other modalities. And therefore, you should early think about them to make the right decisions. so that you later don't have to backtrace, because that can be quite time intense. If you, for example, have a cell line that is instable, it just takes a very long time to redo all of your experiments. All your preclinical data has to be redone with your new cell line. That is quite impactful. That can delay you a year or longer, and that can be a make or break for a company. So you have to be a little bit wise about when you make certain decisions for ATMPs.
And that wraps up part 1 of our conversation with Oliver Kraemer. We've explored the fascinating contrasts between biologics and cell therapy development and uncovered valuable insights about navigating the unique challenges of ATMP development. Join us in part 2 that will drop this Thursday, where we will continue our conversation. Before we go, I want to tell you about an opportunity that could transform your CMC development journey. If you're looking to evaluate and refine your CMC development strategy with precision, I invite you to join my strategy workshop. Book a free call with me to learn how we can help you navigate your challenges with confidence. The links are in the show notes. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.